Thank you very much for such a nice introduction and for the opportunity to present what I have been doing so far. Today, step by step, we will go through the topic of diffusion-weighted magnetic resonance imaging, starting with some basics and then gradually moving on to slightly more difficult ones. And first, let's explain how our brain is built. The brain tissue separates into three types: cerebrospinal fluid, gray matter, and white matter. The cerebrospinal fluid is a clear colorless fluid that is a mixture of water, proteins, ions, neurotransmitters, and glucose, and that occupies the ventricular system and subarachnoid spaces. The gray matter contains the cell bodies, also called somas, of neurons and glial cells, as well as neuronal dendrites and short-range intracortical axons. The long-range axons, often myelinated and densely packed, are the main component of white matter, and it also contains glial cells. At the macrostructural level, axons in white matter are often conceptualized as tightly packed, parallel bundles of myelinated axonal fibers, known as white matter tracts or fiber bundles. White matter tracts are arranged in a complex way, forming a 3D structure through which neurons communicate within and between the cerebral hemispheres, cerebellum, and brainstem. And locally, axonal fibers from different bundles can be arranged parallel to each other or form more complex structures. For example, they can be organized in the shape of a fan. Crossing fibers are the most common configuration. They are presented in up to 90% of white matter regions in human brain. White matter fiber bundles are conventionally categorized into three classes based on their connectivity patterns. The commissural tracts, providing interhemispheric cortical connections, association tracts, which provide intrahemispheric cortical connections, and projection tracts which connect the cortex with subcortical structures such as the brainstem and spinal cord. And now we can move on to the basic of technique that enables us to study the brain macrostructure and microstructure. The principle of diffusion-weighted magnetic resonance imaging is based on the microscopic diffusion of water molecules in the tissue. Molecular diffusion is a random Brownian motion of fluid or gas molecules driven by a thermal energy. Depending on the directionality, two main types of diffusion can be distinguished: isotropic and anisotropic. The isotropic diffusion is also called a free diffusion and is characterized by equal probability of displacement in all directions. It occurs in most fluids, also in cerebrospinal fluid. In biological tissues characterized by a complex structure, the diffusion is mainly anisotropic, so the probability of displacement is different in each direction. And additionally, in the case of nervous tissue, diffusion of water molecules is faster in the direction parallel than perpendicular to the axis of axons. And additionally, two types of anisotropic diffusion can be distinguished: hindered and restricted. And the hindered diffusion occurs in extracellular space and restricted in intracellular space. Diffusion-weighted sequences, which are based on the pulse gradient spin echo technique, are used to obtain the diffusion-weighted signal. The PGSE acquisition work on the principle similar to standard spin echo technique, using two successive radio frequencies, pulses, 90 degrees and 180 degrees. And in the PGSE technique, additionally, two symmetric strong gradient sensitizing gradients are used on either side of 180 degrees pulse. And if the water molecules is stationary, the gradients don't affect the value of the acquired signal, but for diffusing molecules, we can observe a loss in the signal value. And the b-value is the factor that reflects the timing and strength of the diffusion gradients. So the higher the b-value, the stronger the diffusion effects. In order to generate a full data set, first the acquisition of B0 baseline images is performed, which are acquired by running the diffusion-weighted sequence, but with the diffusion gradients turned off. And subsequently, the same sequence is run, but already with the diffusion gradients turned on. The value of acquired diffusion-weighted signal is approximated by the following relationship. The most common diffusion-weighted magnetic resonance imaging technique is diffusion tensor imaging. The diffusion tensor describes the diffusion of water molecules using the Gaussian distribution model. It is represented as a symmetric, positive definite matrix, which has 3 mutually perpendicular eigenvectors and 3 positive eigenvalues. The principal eigenvector determines the main direction of diffusion, so the direction in which the diffusion is the fastest. And on the other hand, the eigenvalues determine the values of diffusion coefficients in each of the principal directions. Schematically, the DTI model can be represented as an ellipsoid with the major axis parallel to the principal eigenvector. Among the measures of diffusion magnitude, the most important one is mean diffusivity, which is the measure of average molecular motion independent on the tissue directionality. The measures of diffusion anisotropy include axial diffusivity, so a parallel diffusivity measure, radial diffusivity, a perpendicular diffusivity measure, fractional anisotropy, which determines the anisotropic diffusion fraction, so the difference between the shape of the given DTI ellipsoid and an ideal sphere, and propagator anisotropy, which quantifies how the given propagator deviates from the isotropic propagator. And the propagator is also called displacement probability density function, and it describes the probability that a molecule in an initial position will end up in a different position after a certain time. In the case of DTI, however, data interpretation is problematic and can lead to many errors. For example, mean diffusivity represents only the overall value of diffusion in a given voxel, but does not contain information about its differentiation in individual directions. And moreover, fractional anisotropy is often interpreted as the integrity of the white matter, but many different factors can cause its changes, for example, cell death. And in addition, the diffusion tensor model contains information only about the main direction of diffusion. So, for example, the crossing fibers can drastically distort the DTI results because we can see that the DTI model seems to be isotropic, but we have 2 directions of fiber bundles. And for this reason, multi-compartment biophysical models are increasingly being used because they assume the schematic geometry of the tissue and thus enable direct estimation of the relevant microstructural parameters. Multi-compartment models assume that the total diffusion-weighted signal comes from 2 or 3 compartments, each weighted by a relative volume fraction. The intracellular space is modeled using axially symmetrical and constrained geometric models such as sticks, cylinders, spheres, and planes. And the extracellular space is most often modeled as an anisotropic Gaussian diffusion, also called a Zeppelin, or a time-dependent Zeppelin, which also takes into account the fact that diffusion is constrained by the outer boundaries of the axons. And these figures shows the correspondence between the model compartments and real tissue components. The most important part of modeling the brain tissue is to determining which microstructural features are relevant to the voxel average diffusion-weighted signal and should be represented in the model, and which one can be omitted without compromising the accuracy. And the first example of multi-compartment models is Neurite orientation, dispersion, and density imaging. The NODDI adopts a tissue model that distinguishes three types of microstructural environment: non-tissue compartment, also called an isotropic compartment or cerebrospinal fluid compartment, intraneurite or intracellular compartment, and extraneurite or extracellular compartment. The non-tissue compartment is the space occupied by the cerebrospinal fluid and is modeled as isotropic Gaussian diffusion, so as a sphere. The intraneurite compartment refers to the space bounded by the membranes of neurites and is modeled as a set of sticks. The orientation distribution of sticks can vary from highly parallel to highly dispersed, and this models the full spectrum of neurite orientation that can be presented in the human brain. And the extracellular compartment refers to the space around the neurites and is modeled as an anisotropic Gaussian diffusion, so as a Zeppelin. The procedure of fitting this model to diffusion-weighted data may be very unstable, and therefore it is necessary to restrict values of some microstructural parameters. However, this may cause incorrect results for some cases. For example, significant differences between the determined values and the values that are actually presented in the human brain in case of some diseases will cause false changes in other microstructural parameters that we want to estimate. So they will not correctly reflect the tissue pathology. The most important microstructural parameters of NODDI model is intraneurite, extraneurite, and isotropic volume fraction, determining the fraction of a given compartment in the voxel volume, and orientation dispersion index, which assess the orientation coherence of neurites. And the second example of multi-compartment models is spherical mean technique. The SMT model assumed that the total diffusion-weighted magnetic resonance imaging signal comes only from two compartments, extraaxonal or extracellular and intraaxonal or intracellular. The extraaxonal compartment is modeled as an anisotropic Gaussian diffusion and the intraaxonal as a set of sticks. And the SMT model makes it possible to bypass two limitations of the NODDI model. First, it does not require the constraints on the values of microstructural parameters. And second, the fit of this model to data is independent of fiber orientation dispersion. So problems associated with crossing fibers do not arise in this case. And the most important microstructural parameters of SMT model are intraaxonal volume fraction and intrinsic diffusivity, which determines the value of axial diffusivity in intraaxonal and extraaxonal spaces. And my first research focused on the use of multi-compartment models in diffusion-relaxometry magnetic resonance imaging. It is very difficult to use only the diffusion-weighted data to separate tissue compartments that have very similar diffusion coefficients, and to use only relaxometry data to separate compartments that have similar relaxation constants. And in both cases, there are theoretical limits to the resolution. The simultaneous use of diffusion and relaxometry magnetic resonance imaging, called diffusion-relaxometry, mitigates the problem of spectral resolution. Diffusion-relaxometry makes it possible to measure multiple magnetic resonance contrasts simultaneously, And combining diffusion with other magnetic resonance contrasts leads to a more detailed quantitative evaluation of microscopic compartments of the brain tissue. Biophysical models are optimized by finding the microstructural parameters values that minimize the objective function, which is the transformed equation of a given model. The brute force algorithm directly built into one of the Python neuroimaging library was used to optimize the NODDI model. And in the case of SMT model, 5 selected algorithms, trust region reflective, Levenberg-Marquardt, Nelder-Mead, Powell, and Newton conjugate gradient were used in order to examine how each of these methods affect the accuracy of the multi-compartment models. And also for the trust region reflective and Powell algorithm, constraints on the values of microstructural parameters were set to biologically relevant. So from 0 to 1 for interaxonal volume fraction, and from 0 to 0.003 for intrinsic diffusivity. And a public available white matter atlas based on the DTI was used to define the brain regions in which the analysis of microstructural parameters were performed. And the microstructural parameters estimated for one subject were registered in a common space of the given atlas, and then 12 regions of interest were selected from them. In the case of qualitative analysis of NODDI model parameters, it can be seen that different parameters react differently to the changes in inversion time, which is also confirmed by the quantitative analysis presented in the form of box plot. The fiber orientation dispersion index is very stable over the entire range of inversion time, while the quality of estimation volume fractions changes due to changes in the inversion time. And this is clearly visible in the box plots, where we can see a characteristic up and down, or down and up tendency. And in the case of qualitative analysis of the SMT model parameters, there is also a clear dependence of microstructural parameters on the inversion time values. And in addition, mostly in the case of intrinsic diffusivity, we can see that only 2 algorithms, trust region reflective and Powell, function correctly. In the case of other methods, numerous bright spots can be observed in the area of cerebrospinal fluid, where the value of the diffusion is about 0.01, which is more than 3 times the values that can be actually presented in the human brain. And this is due to the fact that only for the trust region reflective and Powell method, it was possible to set the limits of the range of the parameters to correct for the brain tissue. And the dependencies which were visible in the qualitative analysis are better illustrated in the quantitative analysis. And for the remaining optimization algorithms, the plots were nearly the same, and they differ only in the range of microstructural parameters. And we can also see a characteristic down and up behavior for both of these parameters. And on the basics of conducted analysis, conclusions can be drawn based on the influence of inversion time and optimization algorithms on the estimation of microstructural parameters. Different microstructural parameters are differently dependent on the inversion time value. For example, the fiber orientation dispersion index is very stable over the entire range of inversion time, while the intrinsic diffusivity and volume fractions show a high sensitivity to changes in the inversion time value, with a characteristic up and down or down and up tendency. and therefore it seems more advantageous to use biophysical models only in the case of high inversion time values, because then we can correctly estimate all of the microstructural parameters and not only some of them. And in the case of tested optimization algorithms, only two of them, trust region reflecting and Powell, can actually be used to fit the SMT model to the diffusion data. And this is due to the fact that only for these two methods, we can set limits on the microstructural parameters values. And of the two methods, the most optimal one is trust region reflective, because it operates in a shorter time, only in 20 minutes compared to nearly 2.5 hours for the Powell method. And now we will move on to my second research. And at the beginning, we'll briefly explain the concept related to tractography based on the diffusion-weighted data. The term fiber tractography refers to any computational procedure by which the anatomical trajectories of white matter pathways can be estimated from the diffusion-weighted magnetic resonance imaging data. And by measuring the diffusion-weighted signal for each voxel, it is possible to assess local fiber orientations throughout the tissue, and then combine them together to infer the trajectories of global white matter pathways that connect distant parts of the brain. And the simplest form of the tractography algorithm assumes that each voxel of diffusion data is characterized by one dominant local fiber orientation. And mathematically, the set of local fiber orientation can be thought of as a three-dimensional vector field, and set of the global fiber trajectories as its streamlines. The numerical integration procedure is starting at the starting point, also called the seed point, and then follows the local fiber orientation over a very short distance called the step size. And this procedure, also known as Euler integration, can reconstruct the entire trajectory of white matter pathways by iteratively executing this procedure. And the presented process of integrating local orientation into a global trajectory is often referred to as a fiber tracking, and the generated trajectory is called a tract. And in turn, a set of tracts is referred to as a tractogram. Depending on the method of choosing the orientation, two main types of tractography algorithms can be distinguished: deterministic and probabilistic. The deterministic algorithms follows the estimated local orientations step by step until a certain termination criterion is met. And the tracking procedure initiated in a particular voxel always follows the same path. And deterministic algorithms are considered specific and insensitive. They can correctly reconstruct the known paths of the fiber bundles, but at the same time, they can skip generation of shorter trajectories. Probabilistic algorithms, on the other hand, generate a large set of potential trajectories from each seed point, and they are referred to as sensitive and non-specific. The trajectories between two regions of interest in the brain can always be reconstructed, but this results in a large number of false positive trajectories. The most important factor that determines the tractographic algorithm propagation is the diffusion model used to estimate the local fiber orientations in each voxel of the diffusion-weighted data. Individual models provide different information about the brain structure and are differentially sensitive to the presence of more than one fiber orientation in the voxel. The diffusion tensor model is most often used, but it doesn't enable the detection of crossing or fan-shaped connections. And due to the presence of numerous areas of crossing fibers in the brain, the methods based on constrained spherical deconvolution or fiber orientation distribution are becoming more and more popular. Regardless of the diffusion model used, tractography has a number of limitations that can reduce the accuracy of the reconstructed connections in terms of biological architecture. And for this reason, it is beneficial to use prior knowledge about anatomy of the brain and structural information to guide the tractography procedure. And however, generating only a simple binary masks, for example, defining the white matter regions in the brain, makes very little use of the available anatomical information. And anatomically constrained tractography is the method that enables more effective use of the information provided by segmentation of anatomical data. And instead of defining only a binary mask, the biological properties of individual brain tissue types and the nature of white matter axons are used to define the termination site of tractography procedure and tract acceptance criteria based on the biological plausibility. And 6 anatomically based prerequisites can be defined in the ACT method. And for example, if the tract penetrates the cortical gray matter, it reaches an appropriate endpoint and is accepted, while if the trajectory penetrates the area filled with cerebrospinal fluid, it is rejected. And in addition to using anatomically constrained tractography, it is also possible to remove excessive and non-anatomical trajectories from already generated tractogram. And in this context, the concept of tractogram filtering emerges. Filtering methods are designed to match the correspondence between the signal predicted by a streamlines passing through a given voxel and the actual diffusion-weighted signal in order to achieve a correspondence between the local density of reconstructed connections and the local density of real fibers. And initially, the algorithms used enabled this to be done by selecting a subset of tracks that best fit the diffusion-weighted data. But later methods enable to estimate weights for each streamline, and the weights represent the effective cross-sectional area of each streamline, which can be used as a direct measure of connectivity density. And in my second study, I used the tractography algorithms to map the main trajectories of white matter pathways and to analyze the variability of microstructural parameters along them over the years. The natural processes of brain maturation and aging are associated with numerous changes occurring in the brain structure. Brain aging involves multifaceted changes in white matter integrity, including the demyelination and axonal loss. And among other things, the demyelination process is associated with the loss and functional dysregulation of oligodendrocytes, whose degeneration increases with age, as well as the loss of myelin proteins. And so far, numerous studies on the brain development have been conducted, but they were mainly based on the analysis of changes in volumetric features and microstructural parameters, but mostly in the case of regions of interest and tract-based spatial statistics. And these methods have many disadvantages. The regions of interest are prone to partial volume effect, and the tract-based spatial statistic is based only on the FA skeleton, and therefore has limited anatomical specificity. And in my research, I used a tractographic approach that allows to avoid these problems and analyze changes in microstructural parameters along the trajectories of white matter pathways. Based on the diffusion-weighted data, fiber orientation distributions were determined, which were then used to generate the whole brain tractogram by applying the second-order integration over fiber orientation distributions algorithm with anatomically-constrained tractography framework. And the obtained tractograms were further filtered by using the spherical deconvolution informed filtering of tractograms in order to remove the excessive and non-anatomical connections. The obtained tractograms were registered in the common space of HCP 842 Tractography Atlas, containing the standard whole brain tractogram and 80 selected white matter pathways. And then 4 fiber bundles were selected from them by applying the Reco bundles method. And these bundles were arcuate fascicles, corticospinal tract, inferior longitudinal fascicles, and uncinate fascicles. And also microstructural parameters based on DTI, so fractional anisotropy, propagator anisotropy, mean diffusivity, axial and radial diffusivity, based on the NODDI model, so intraneurite, extraneurite, and isotropic volume fraction, and on SMT model, so interaxonal volume fraction and intrinsic diffusivity, were also determined from the diffusion-weighted data. The selected set of tracts were divided into 100 segments based on the distance between individual trajectories points and points on the centroid. And in these segments, the variability of parameters was examined. A Poisson regression model was used to analyze the trajectories of mean measures changes across the lifespan, because it was shown in the literature that it most accurately reflects the nature of changes occurring during the brain maturation and aging. And already in the case of qualitative analysis, as several characteristic dependencies can be observed, the trend of changes in a single microstructural parameter is not the same over all of the fiber bundles, but in subsequent age groups, the nature of changes in parameter values is nearly the same, but the range of values changes. And in the case of right arcuate fasciculus, the fractional anisotropy shows the highest values in about 1/4 and 3/4 of the pathway length, and the lowest in the central part. On the other hand, in the case of radial diffusivity, high values are characteristic for one end of the bundle, and for the other, the values are the lowest. Extraneurite volume fraction of NODDI model shows higher value at the end of the bundles and lower in the middle part. And the interaxonal volume fraction of SMT model shows an opposite trend. And in the case of corticospinal tract, fractional anisotropy, radial diffusivity, and interaxonal volume fraction of SMT model shows higher values in the middle part of the bundle and lowest at the ends of the white matter tract. And extraneurite volume fraction of NODDI model shows high values in the upper part of the bundle, while in the lower parts, values are the lowest. And the dependencies visible in the qualitative analysis are confirmed by the quantitative analysis. The trend of changes in microstructural parameters is similar for the single fiber bundle in all age groups. And moreover, the changes in the values range observed with age is more visible. And in the case of radial diffusivity, the values are the lowest in the middle age group, and interaxonal volume fraction of SMT model shows an opposite trend. So in the central age group, the values are the highest. And in addition, the analysis of trajectories of microstructural parameters showed regionally differentiated nonlinear changes over a wide range of age. And in the case of fractional anisotropy, these changes are in an inverted U shape, with the maximum occurring mainly between 25 and 30 years. And the rate of changes observed in adolescence and early adulthood is greater than later in life. And the radial diffusivity shows an opposite trend, that fractional anisotropy, and the minimum occurs a bit later in life, mainly between 28 and 33 years. And in the case of extra Neurite volume fraction of NODDI model, the nature of changes is the same as in the case of radial diffusivity, but the extremum occurs later in life, mainly between 30 and 35 years. And the intraaxonal volume fraction of the SMT model shows the same trend of changes as fractional anisotropy, but the maximum occurs in the same age range as in the case of extra Neurite volume fraction of NODDI model, so between 30 and 35 years. The qualitative and quantitative analysis proved that the nature of changes in a single microstructural parameter is not the same over all of the fiber bundles, but it retains similarity for the same white matter pathway from the right and left hemispheres. And this means that the corresponding regions of both hemispheres are characterized by a similar tissue microstructure. And this microstructure varies between the different areas of the brain. And quantitative analysis additionally proved that the trajectory of median value changes are the same in subsequent age groups, and only the range of values changes. So the study showed no variability in the rate of maturation in individual parts of fiber bundles. Analysis of the mind fiber bundles showed regionally differentiated nonlinear trajectories of parameter changes, the extremum of which mainly occurs between 25 and 35 years. The inferior longitudinal fascicles shows the earliest, and uncinate fascicles the latest transversal point, and it can be connected with their functions. The inferior longitudinal fascicles is most likely to develop the earliest, because it is responsible for the basic processes such as facial recognition and visual perception. And the uncinate fascicles is mainly involved in the linguistic functions, hence its slow maturation. And now I am working on some analysis to my PhD study, and they will be partially connected with what I have been doing so far and what I have presented to you during these seminars. And now I am focused on the determination of microstructural parameters of brain tissue via deep learning models and connecting it with a federated learning approach. And in the future, I want also to analyze the variability of microstructural parameters across the lifespan, but using much more data than I show you in this presentation. So probably I would be also working with the data harmonization problem. And maybe in a year or something like that, I will have the opportunity to also present you my results. And the presented researches are were the part of my engineering thesis and master thesis, multi-compartment models in diffusion-relaxometry, magnetic resonance imaging, and mapping the properties of white matter pathways of the aging brain using the diffusion magnetic resonance imaging. And the supervisor of these two theses was the Tomasz Pięciak, PhD. And that brings me to the end of my presentation. Thank you very much for your attention. If there are any questions, I will try to answer them.